Well, welcome to our pathology lecture. We have a really terrific speaker tonight. Dr. Kimberly Cochran is a pathologist with Texanium AgriLife, and she's been doing this for me for quite a while, and I appreciate her, and I think you're going to learn a lot from her lecture. So please welcome Dr. Kimberly Cochran.
Hello. So I am located in Uvalde, Texas. So I'm a good bit like kind of northwest of you guys, but a lot of our pathology issues are going to be pretty similar. And I have made, as Kevin mentioned, several trips down there. So I kind of have a small grasp on some of the more specific issues that you guys deal with. So we will go ahead and get started. You should be able to see my screen, but I'm going to switch you to full screen. So first we're going to cover plant health and plant disease. Then we're going to talk about and look at plant disease symptoms and signs and some examples of both of those. Then we're going to go over specific groups of plant pathogens and some disease examples, disease diagnostics and exercises, disease management tools, and resources and other info. So you guys do have an abbreviated class. We will have a short break, but it will be shorter than my other class breaks. Typically, this will be a 3.5 to 4 hour class. So we're going to hit everything we need to hit, but we're not going to have a lot of bonus time. All right. So what exactly is plant health? It's kind of like a buzzword nowadays within agriculture and plant sciences in general. So there are a lot of different definitions, but in a nutshell, it's when a plant is free of disease, stress, or injury and can reach its full potential in terms of yield or other production measures. So that might be pounds of cotton per acre, or it might be bushels of corn per acre. It could be number of cut rose stems in a commercial flower production. It might be how many tomatoes per plant you got out of your garden. Or in nature, it could be more of a measure of how many successful offspring did that particular individual or population of individuals have. So our scope of success can shift, but generally for the purposes of people, we're talking about like yields and harvests and aesthetic qualities. All right, so why exactly is plant health important? Not to toot my own horn, but plant health is really critically important to kind of some of the underlying core like supports to society. So focusing on maximizing our plant health can help maintain the security and stability of our food and feed and fiber production and fuel. I know I only have food on the slide, but there's actually a lot of other things that our crop agriculture feeds into, literally. So that's a really critical component to maintain a stable society, because if we're significantly lacking, and I don't mean just like, oh, you don't have your particular brand of shredded wheat at the store. I mean, like, there's truly nothing to sustain you, and it's a true famine situation. The pillars of society crumble very quickly. So we're not really used to that here in the Western Hemisphere in the past several generations. We don't really have hunger due to lack of availability of product. We might have hunger due to lack of resources to obtain that product, in other words, money, but the actual food product is plentiful. So anyway, that's kind of, I don't want to sound overly self-important, but it is kind of a big deal. Also, we really focus on maintaining our farmers, and that includes direct cost of disease management, so like fungicides, fuel to apply those, hourly workers to apply those, wear and tear and maintenance on their equipment. Also, we look at yield loss and reducing the impacts of that in both quantity, so the amount, and the quality. So let's say we have a field of corn. Sometimes we might lose 20% or 10% to disease. Well, that's a really easy thing to measure. A thing that's a little more difficult to measure sometimes and quantify in terms of financial loss is quality reduction. So let's say we have a load of premium corn and the farmer takes it to the grain elevator and they get like premium prices, next farmer has the same pounds of corn in their trailer and their truck that they're taking, but it is a lower quality due to, say, fungal pathogens that have invaded the corn kernels, and it's reduced the kernel starch composition or weight per number of kernels, for example. So they'll get less money at the grain of an elevator. So it's not just actual pounds produced, but quality of pounds produced as well. Now, I know there's a lot of preconceived notions about corporate farms, and I'm not here to preach or give you absolutes. And I can't say that there are no corporate farms that grow row crops or vegetables, because I haven't been every single place ever. But I've been over a good portion of Texas and a good portion of Arkansas. And I have never worked with a corporation that owns land and farms. I've worked with many farmers that own land and farm. Now, that farmer may run it as a business, like L&L Farms, for example, is owned by the LaFear family, and the business is L&L Farms. And they might contract with HEB, or back in the day, it was Del Monte, or some of the other companies like that. But the family is the one that actually owns it. So, I've had people ask me, well, what about, when I drive by these cornfields and wheat fields, I see these signs out that say like DuPont 5522 or something, or Pioneer signs. And the thing is, those are not necessarily ownership signs. Those are often markers for plot trials. So it can lead to some misinformation that's floating around out there in the world. And as an extension professional, that's part of my job, is to clear that up. So that's just been my experience and my little bubble of life. But that's just my 2 cents. So we really strive in plant pathology to help reduce the risks of these losses for individuals and industries. Like, for example, I work with all the spinach farmers here in the Texas Winter Garden. I just don't pick one family, but there's like 30 families that grow spinach, and together they're the Texas Winter Garden Spinach Producers Board. So I work with the producer board. So we work with groups like that, but our end person we are serving is the public, and it's families who are just trying to make a living. All right, so I'm off my soapbox. What is plant disease? There are a lot of different definitions that float around. And generally, it's when there's a disruption of a plant that interferes with the normal structure, function, or growth, and results in symptoms. Now, conversationally, when we say disease, a lot of times we're using the broad sense of the word, and that can include anything wrong with a plant, like disorders from like chemical burn, impact injuries, which are mechanical impacts, heat, drought stress, freeze damage, things like that. Now, in the narrow sense, when we say disease, that's referring to things on the yellow side, which include things that are from living or biotic issues. And these include our main categories of pathogens like viruses, viroids, bacteria, fungi, nomycetes, parasitic plants, and nematodes, insects and mites, and other herbivores like deer, porcupines, critters of all kinds. But when you hear plant pathologists say plant disease, we're really talking about the ones that are there in the green. So pathogen or microbe-caused issues. All right, so what plant parts can be affected? Unfortunately, all of them. And we see a variety of symptoms ranging from wilts to leaf blights to spots, fruit rots, root symptoms like rots and malformed root tissues. And generally, these will be categorized into soil-borne and foliar pathogens. And that's where we primarily see the point of infection and the initial symptoms of these issues. So that's how you can kind of conceptualize that. Things that live in the soil and infect through the soil, those are soil-borne, or infect through the roots, I mean. And then things that are foliar are going to be primarily infecting all those above-ground tissues. And they don't really live in the soil on their own without a plant host. So types of pathogens, again, just to recap, are fungi, bacteria, viruses, and nematodes. Notice I've condensed this list a little bit just for the purposes of this class, because it's an intro class. And the biggest group that I see most often is fungal pathogens. And that's in bold to help you remember that. So you will get a handout of this. You'll get a PDF copy. So don't worry about trying to scribble everything down. I know it's going really fast because you do have that condensed time slot for your class. But again, you'll get a copy. So now in terms of the order of the topics we'll cover, I'm going to save the fungal group for the last because that's how it works the best to lay it out for breaks. So, all right, so now that we've covered some of the basics, what are the main components required for a disease to occur? Because we have pathogens, right? We have plants everywhere, but we don't have disease every second of every day. So a good way to visualize this is the disease triangle, and we have to fulfill all 3 points to have a disease event occur. So we need a susceptible plant host, we need a viable plant pathogen, and a favorable environment for that pathogen. And all 3 of those things have to be present at the same place at the same time for a disease event to occur. Now, a lot of updated texts will include a 4th point and make it a pyramid, and that will be time, because disease events need time to progress. They don't just go from, the plant's in perfect health, 0 problems, to bam, it's dead. We need time for that severity to increase, for those microbes, the pathogens, to colonize and reproduce and spread and continue their epidemic cycle. So time is a really big critical thing. And that's a big clue about being able to tell the difference between a non-living cause versus a living or biotic pathogenic cause. Abiotic things happen like bam, but pathogenic or biotic issues require time to develop, and they will worsen with time. So I'm going to plant some seeds along the way for some topics we'll cover later on. But notice when we disrupt 1 or more points of this disease triangle, it's no longer fulfilled. So kind of tuck that away in the back of your mind, because we're going to revisit that when we talk about disease management. Okay, so also along the way, you'll have these little note boxes and some vocab tips and things like that. That's just to help you in your note taking when you get the presentation copy. So you'll have those as well. All right, so we're going to go over a very basic generalized disease cycle. We talked about what's required for a disease event to occur. So what happens during that disease event? That process I talked about that takes time. So we'll start at the very top just to have a good place to start. And that's the overwintering or overseasoning or resting stage of the pathogen. I don't personally like the term overwinter because it implies it has to be cold. It does not. It just means whatever season is least favorable for that pathogen, it's at rest. So this can occur in the plant tissue itself, in an insect vector. In some cases, plant pathogens are taken into the insect's mouth parts, and it can walk around with the pathogen in its mouth and upper gut for a while. It can also occur in plant debris. So like, say, an old tomato that was diseased, fell to the ground, and that hung out for till next year. That would be a good example of debris. It can also occur in the soil itself. In some cases, not all pathogens can survive in the soil on their own, but a good portion of them can. And those are soil-borne pathogens. Sorry, I need to mute myself. I always forget to mute myself, which is the worst, to have your phone going off in your own lecture. There we go. Okay. So we have a resting or overseasoning stage. Now let's say a plant host becomes available, and it's springtime. We have a new plant growing there at that location and where the pathogen is at. And we have favorable conditions. It's nice, 75 degrees, moist, hypothetically. So we are fulfilling all 3 points of the disease triangle. We have a plant host, a viable pathogen, and favorable conditions. So at that point, the infection process will start. And during this kind of like cat and mouse game, or like a little dance that the pathogen and the plant do, the pathogen is trying to gain entry to the plant and then overcome its internal defenses. And if that is successful, then the pathogen will start colonizing the plant tissue and then start causing some disease symptoms like wilting, yellowing, stunting, things like that. And if the environment continues to be favorable, the pathogen will start reproducing and spreading. And that can happen in a variety of ways. So some go back into kind of a resting state, but many foliar pathogens in particular go straight back into finding a new plant host. And if those conditions are still favorable, then they'll just loop right back into the infection process and continue on to reproducing and spreading again. And now this is called a secondary cycle. And again, I don't like that term secondary because when I first started out, I really struggled with it implies there's only two, but that's very misleading because it's actually more like compound interest. And some newer texts actually call it compound interest or compound diseases because it can happen over and over and over and over again as long as the conditions are favorable and you have adequate plant host material. So if we think on that and think about how like compound interest accumulates, that's a really steep increase curve, right? It's like an S curve. It's the same thing with these pathogens that have these compound cycles within a single season. And they can be really difficult to manage if you wait too long until they've reached that steep part of the S curve. So they're just notoriously difficult with management techniques, and we'll cover that a little more later on. I don't want to get ahead of myself. All right. Any questions before we move on to disease symptoms and signs and looking at pictures of those? And if you think of it later, you can always put it in the text or you can always email me. That's fine too. Okay. All right. So symptoms are changes in the plant in response to the pathogen, like wilting, yellowing, tissue death, stunting, which is when it's smaller than it should be, and galls, which are malformed growths. Signs are direct evidence of the pathogen, like actual pathogen tissue. This includes fungal tissue, like reproductive structures or sporulation, and bacterial ooze. So here's an example of signs versus symptoms. This is a corner of a cabbage leaf. That was my first cabbage sample ever. And notice our symptoms are yellowing or chlorosis, necrosis or tissue death, and it looks like it was pretty wilted when it came in. Now I looked closer and saw this white cottony stuff and these little black flecks. Now this little panel on the right is just that picture but cropped and zoomed in. And you can see these little black flecks a little more clearly. So these are fungal reproductive structures on the black part, and the white is fungal somatic tissue or the body of the fungus that's colonizing the plant. And those are both signs. So in this picture, we have both symptoms and signs. And I've included some little reminder arrows for your notes as well. All right, so some common symptoms that I'm sure you've all seen, whether you knew it or not, are leaf spots, which are yellowing and dead spots, like you can see in this top right picture. And leaf spots start out as discrete individual spots. They can merge together into more of a blight and cover most of, or not all of, the leaf. These are usually going to be caused by fungi, bacteria, and viruses in that order of prevalence. General yellowing, in contrast, general full plant yellowing would be something that affects the roots more than likely, like say root knot nematode or fungal root rot. So just kind of like think about how that would look very different. And we'll look at some stunting pictures later on in the presentation. Here's another shot of a leaf spot on peanut in this case. So we have tomato and peanut. And notice that these spots look kind of similar. The pattern on this one on the left is a little more clear. But fun fact, these are actually caused by the same pathogen or the same genus of pathogen called Alternaria. And this is a very common issue that causes a lot of leaf spots in a lot of different plants. And this is a good example of how a particular pathogen can sometimes be broadly infective over many different types of hosts that are highly varied. Like tomato and peanut are not closely related, right? One is a solanaceous crop, the other is a legume crop. So they're not really, they're not like comparing tomatoes to eggplant or something like that. These are pretty distantly related. So some pathogens are very broadly pathogenic to a lot of different things, which can make them extra difficult to control. All right, so here's another photo to kind of compare and contrast. And this one is of oak wilt on a live oak tree. And Notice that the yellowing starts on the veins and then it progresses to tissue death and then moves outward away from the veins. So that looks very different from like leaf spots that are discrete little spots that have like a yellow halo and then tissue death in the middle. This has a very striking contrast. And a little vocab tip box for you guys. All right, some more common symptoms include canker and dieback. And this is often fungal or bacterial and cause more often than not fungal. I see this a lot on like woody ornamentals and some trees as well. And it's exactly what it sounds like. It dies back down the cane or the branch and progresses closer and closer to either the crown or the trunk, depending on what we're dealing with. And the line here is the canker line, which you can see on this right panel. Yeah. So here's another shot of dieback. It's going down the twig and it's progressed into the larger branch on this. I want to say this is a apple tree, maybe. It's either apple or no, it's apple. Yeah, that's right. So anywho, this is a better shot of where you can see the canker doesn't completely circumnavigate the branch. It's more like a lesion type pattern like this. Instead of being girdling the branch, it's just a more discrete circle. So cankers can either look like this, where it's all the way around the perimeter of the stem, or more like this, where it hasn't progressed all the way around yet. So they can vary. Galls are malformed growths, and these can occur on roots or above ground tissues. They can be caused by fungi, bacteria, or nematodes. More often than not on roots, it's going to be due to nematodes. In above ground cases, most of the time when I see it, it'll be fungal in nature. Here's another look at some galls. So we often see these on trees, unfortunately, and it can occur on the main trunks. It can occur on the crown area. And a little fun fact and silver lining of galls is that burlwood is actually caused by these malformed growths in the wood. And then we take it, harvest it, cut it and polish it, and it looks pretty. So at least there's a little something that comes from the demise of these plants. And it's highly valued for things like inlays and fancy pens and other little doodads like that. So neat little fact there. All right, so some other symptoms are root and crown rots. I see this a whole lot when people are starting seed. And here on the left panel, you see a normal healthy root. In the middle, that thing's a goner. It's really badly rotted, very discolored. It looks really squishy, which it would be if you were to, actually touch it. So that's a pretty severe case of damping off or root rot. Now, this panel on the kind of center, but it's like the right side of that little portion. Notice that we still have some decent amount of roots. We have decent root development, but they are discolored. And this is a really good illustration that plant disease is not always an all or nothing thing. It's a spectrum of severity. So your outcome is not always going to be an all or nothing situation, like perfect health or complete death. So that kind of in the middle or median seedling, it would probably live unless it had some other major stressors during its season, but it would probably live. It just might not, produce to its full potential and have the most wonderful beans ever, but, it makes some. So plant disease is a spectrum of severity. Another issue I see a lot is crown rot, and I see this a whole lot in houseplants and in succulents and cacti. And these are typically, both crown rot and damping off, going to be caused by fungi, oftentimes a complex of more than one, or fungi with another group called oomycetes, which we'll touch on later. Occasionally with nematodes, especially in row crops. I used to see that a lot, where we would have some seedling disease paired up with nematodes, and it would just add insult to injury and be what's called a disease complex. And they'll be significantly worse together than either would be on their own. Occasionally, in really wet conditions, when it's really warm, I'll see bacterial damping off, but that's pretty uncommon. The only time I've seen that is in some flooded or recently flooded soybean fields in Arkansas. And when I say flooded, I mean like the river flooded. And it was like 95 degrees after that for 2 or 3 weeks. So these newly planted bean fields had a lot of bacterial rot. So, uh, okay, so we're going to talk about plant pathogens and disease examples by group. So we're going to cover bacteria, viruses, nematodes, and then fungi. So bacteria, as we all know, are single-celled organisms, and they are everywhere in the environment. But the good news is that only some are pathogenic to plants. Most that cause disease in plants will not make people sick. However, if someone is immune compromised, then there might be some gray area there. So there's a really good publication put out by Iowa State and their extension service that has some more information about that. It's also really important to note that some bacteria that are pathogenic to people but harmless to the plant itself can survive on plants. And this includes E. coli, Listeria, Salmonella, and several others. I'm sure you've all heard of these. It's the reason we get like lettuce recalls and tomato recalls. So as my public service announcement, please just be mindful if you have animals on your property or your adjacent neighbors do and you're close. Things like pigs, goats, sheep, cows, chickens, even horses, but I think they're maybe a little lower risk. I could be wrong about that. But basically, if you have any animals that could get into your garden and growing area, just be aware that you need to wash all your produce very, very thoroughly. And also be mindful of water runoff. So let's say the animals don't get into your garden, but you have a really heavy rainstorm like what's coming up in our area of Texas, and then you have runoff from one area of the property or your neighbor's property into your garden. And so anything higher elevation like the animal pen, if that runs off into your garden, voila, you have a bunch of animal poo in your garden. This also extends to fresh fertilizers. I know everyone likes animal poo for fertilizers, and it can be great. However, do not use fresh because A, it'll burn your plants. B, it's rife with bacteria of all kinds. So only use composted or otherwise heat treated manures of any kind in your garden. And I would also encourage you to be extra aware of these topics if you are selling your produce at, say, like the local farmer's market or something like that. But in a nutshell, always wash your produce really well and be aware of where the poop goes. That's my PSA. So, and I figure if it prevents one person from being sick, it's worth it for me to get on my, as my husband likes to call it, poop soapbox for a few minutes during every lecture. All right. So how do bacteria get into plants? They're pretty crafty, but they're not really powerful on their own. So they can't like force their way into the plant. So they rely on openings. Plant pathogenic bacteria are going to be mobile. They'll have little flagella or cilia, and they can actually move through a little thin layer of water or be splashed from one place to the next in some water. So they can go in through natural openings like stomata and hydrathodes, which are shown here in these two photos. And they can also get in through any rips or tears or insect feeding sites that puncture or chew along the leaves. So any opening, whether natural or made by a living being or mechanical injury. So how can we see bacterial sign if each bacterial cell is so small that it's microscopic? Well, obviously we cannot see individual bacteria without a really fancy microscope, but we can see them in mass. So that's what we're looking at on that upper photo. Those kind of gross looking droplets, those are actually bacterial ooze droplets, and each one contains millions of bacterial cells and metabolic products that they have made, as well as probably some plant juices. But this is called exudate collectively, and it is a key feature for a lot of foliar pathogenic issues. And like if I were looking in a field and I saw that well, I would know that was a bacterial issue, but I would certainly want to do some confirmation diagnostics, particularly if it was a high stakes situation. Okay, so for the purposes of class, I'm kind of trying something new this past year or so on dividing up bacteria a little differently. So for this class, we're going to group them in two informal groups based on symptomology and where the infection occurs. So we will talk about vascular bacterial pathogens, which you can see an example of on that top photo with where we have bacterial wilts of squash. And then we'll talk about leaf spot bacterial pathogens. And they're exactly what they sound like, leaf spots. The thing with bacterial leaf spots is they often start out very small, like the size of a pinhead, and they can grow a little larger, like you can see on these pepper leaves down here in the bottom. But they will almost always start out as tiny, tiny little spots. And that's kind of a red flag for me when I get a sample in and I see tiny spots that have, and then different stages of infection where the spots get larger and larger and merge together. Also, things like a really dark, kind of moist looking, what we call water soaked center on the lesions, or in some crops will have kind of a very dramatic red perimeter around the edge with a really heavy yellow halo. So what I would do in the lab is take little tiny cross sections of those leaves and put them under the microscope and watch for bacteria to pour out from the leaf. And we can also do that with fruits. The symptoms look very similar, but very rarely have I seen bacterial spot in fruit that was not also there in the leaves. So, all right, bacterial wilts will pretty much will always have this really water-soaked, kind of moist-looking limp appearance, and it results in death of the plant. The early stages are going to first be noticed midday, where the plants will be wilty even though you water them. So no matter how much you water, it will never perk up. These unfortunately do spread really easily by water splash. So the more you water it, the more you're spreading it. And it's really, really easily spread by wounds. And I see this a lot when we have like phloem feeding insects or something like cucumber beetles. And when we have a really heavy infestation, they'll just go make all these little wounds all over the plants. And it's just like a smorgasbord for these bacteria to just get in and have a field day. So with wilts, though, there are some tips and tricks that we can do to look for or confirm suspected wilts. So both these photos on the bottom, there's clearly something wrong with these plants, right? So it would be recommended that you remove them either way, no matter what the diagnosis is. But what I would do is remove those plants, do some kind of visual inspection for the first steps of diagnostics, and I would split that stem lengthwise, like in that top photo, and I would look for this kind of rotted out chocolatey brown appearance. That's characteristic of bacterial rot. There are some fungal vascular rots that look a little different, but they're not going to be as brown as this, in my opinion. Now, that's a little harder to do with cucurbits just due to the type of stem they have. So with that, honestly, doing a clean cross section with like a razor blade and also doing some more advanced diagnostics is usually more informative. So for prevention tips, focus on insect injuries because, again, that's just making a ton of little avenues for all those bacteria to get in. Reduce your water splash and canopy humidity. Say switch to drip irrigation if you can. Reduce the density of your canopy. So like these tomatoes in that bottom photo, they're planted too close. So you're going to have a really humid canopy. They're not pruned up very well. So just do things that you can to promote airflow and get things nice and dried out. Remove infected plants. Practice good sanitation. So when you do pull them, don't just lay them down. You need to like take them away and dispose of them. Either burn them if you don't have a burn ban or put them in the trash. You can rotate your crops for a minimum of 1 year. I like to do 2. And make sure you don't have any volunteers that have grown from some dropped fruits or something, and no weeds, because there are a lot of weeds related to a lot of our vegetable crops that can harbor not only the bacteria, but the insects that aggravate this situation. All right, so if you have some samples and you've cut them lengthwise and you're still at a loss, another really easy way that you can check for bacterial wilts is, well, there's actually two ways. There's the cut stem method and the cup method. So with the cut stem, you just take the stem, cut it with a clean razor blade, put it back together, and slowly pull it apart about 1/4 of an inch and see if there's any really, really slimy, thick stringing that happens. And this will be much thicker and slimier than the sap. Sap will be watery by comparison. So this is going to be more like mucusy. And that's compounds that the bacteria have actually made. And that's a diagnostic feature. Another really easy method is the cup method or bacterial stringing method. And you cut a stem, put it in a cup of water, obviously clear cup, and observe for these little tendrils that will come flowing out. So I actually have a little, nope, wrong one. There we go. You should be able to see it. Is it working?
Yes.
Okay, excellent. All right, so this is real time. It's not sped up, and you can see these just like little tendrils of white goop coming out, and that's not a technical term, but it's bacterial streaming coming out from the stem. So it's super duper easy. It won't always look this dramatic. You might only have 1 or 2 little tendrils coming out, but it is visually obvious and doesn't require a lot of expertise and additional training. Okay. All right. You should be back on the presentation. Can you guys see that okay?
Yes.
Perfect. Okay. So now that we've kind of touched on bacterial wilts, there are some other vascular associated bacteria that don't really cause a classic wilt symptom. And one that is highly relevant for you guys with where you're at is citrus greening. So this is caused by bacteria that infects the phloem of citrus trees, and it does eventually kill the tree. It goes by a couple different names, Liberibacter, Huanglongbing, or HLB. And the most common term here in the US is citrus greening, though. It is spread by an Asian citrus psyllid, and it is also spread by humans moving plant material like budwood, leaves, even fruits, let's say. Basically any plant material whatsoever. So this little insect vector is very, very small. There's a finger in relation to it. So it's like this, smaller than the tip of a pencil lead. It's so tiny, which makes it really difficult to find in like USDA inspections and things like that. And it can get in on like in pallets on fruit shipments from South America and things like that. So it's super duper tiny, and it spreads this bacteria very well. So let's say we have a beautifully drawn insect over here on the left, this little gray dude, and it has bacteria, the Liberibacter bacteria, in its mouth parts, and it lands on an uninfected leaf. And it's picked up this bacteria from feeding on an infected tree in the past. But it lands on a new uninfected leaf, and it decides to feed with its little straw-like mouth part called a proboscis, and it pierces the leaf and then starts slurping up nutrients. And the bacteria are transmitted down the proboscis or mouth part into the plant. And there, the infection process starts. The pathogen works to overcome the plant defenses, which are chemical defenses. Plants don't have immune systems like people with white blood cells and things like that, but they do have more of like a defense system that operates with chemicals instead. And it tries to recognize pathogens and defend itself with chemicals, in a nutshell. So with citrus greening, that is never successful. The plant's never successful because we don't have any varieties of citrus that are able to do that successfully. So the bacteria do eventually reproduce and proliferate within the vascular tissue of the plant. And you can see that in this middle bottom picture. That's the vascular tissue. We're looking like if you had a straw, like this one, and say this is the vascular bundle, right? And we're looking down at like this on that bottom middle picture. So all the bacteria are clogging up that vascular tissue. and they're also secreting toxins and things like that. And it results in some pretty significant symptoms and death of the plant because its vascular tissue is not able to work correctly. So some common symptoms include small, bitter, misshapen, discolored, just weird looking fruit. Also raised or thickened looking veins, an asymmetrical modeling of the leaves. And modeling is just irregular coloration. And you can see that on the bottom right picture where it's too light, almost yellowish, and then kind of a more regular green. And there's no even pattern. It's very irregular. And this is the most common issue that we see here in Texas. So currently, we don't have any cures or really therapeutic treatments for infected trees. So prevention of spread is super, super important. Some people do spray for vectors, but again, those little vectors are so tiny, it is very difficult to make sure you have really good coverage to make sure you get all of them. And also, let's say we spray 2 days ago, and then there's this tropical storm coming in, like tonight, tomorrow, and the next day. Well, guess what the tropical storms bring? These insect vectors. Yeah. So it's a challenge. Even if you spray today, a tropical storm or even just normal wind can bring these little guys in and infest your plants, in a week's time. So that's really challenging. So we really focus on early detection and removal of the tree to help limit the spread of disease, because the longer an infected tree sits there as it's progressing toward dying, the longer it has to be a point of inoculum for trees around it. So we want to yank that out as soon as possible. It's really unfortunate for the grower, or the homeowner, and they're expensive to replace, but it is what it is. I hate to say that, but that's the best we can do for now. So there's a really big focus on protecting areas that are growing citrus that don't have this yet. As far as I know, and this is not up to date, I openly admit that, but as of a couple years ago, this had not made it to California yet. I don't know if that's still true. But, like Florida had it before Texas had it. It eventually crossed the Gulf, came into Texas, and we got it around 10 years ago, I'd say. And it's just been really difficult. In Florida, it's been such a problem. They've lost millions and billions of dollars in production and millions of trees. And as you know, with citrus trees, it takes years to get that back into production. And even if they were to replant, they would just be reinfested with psyllids and the pathogen. So anywho, quarantining is super important to limit the spread, and early detection is our current frontline. DNA screening is really sensitive, but you can get a false negative through no fault of the technician or no running the test, and no fault of the test itself. It's just random chance. So let's say on this tree in the background, I sample from the bottom branch, the top branch, and then on like the left side of that tree in the middle. Well, if I didn't happen to sample a branch that was recently infected that had not spread to the whole tree yet, I could, conceivably miss a positive, but just simply due to sampling error. So that can be a really big challenge with some of these issues, with these really impactful diseases. So there's been some interest in using dogs with scent work to find infected trees and remove them earlier. The main challenge, though, is it's really expensive to train these dogs, and it takes a year or two to get a working dog raised up and trained. And then you have a limited number of years that they can actually work, maybe 8, at best 10 years, depending on the breed, and then they have to retire. So it's an issue just due to the inputs required. Currently, our main go-to person is Dr. Femi Alabi at the Westlaco Center. You guys are like halfway between Femi and myself, so feel free to reach out to him if you have any questions about citrus greening, because I will openly admit he knows more than I do about it. All right, so this is an older map, but there is a Texas quarantine map available online, and it'll show you by county What areas are quarantined, and this applies to both plant material and insect vectors, particularly within shipments. Now, you guys are probably not receiving a lot of commercial shipments of citrus material. However, I'm well aware of the roadside pop-ups, and we all know what I'm talking about, right? Where people will have a van of plants and they just pop up for the weekend at an intersection and they sell a bunch of plants. That's an area of concern in terms of disease transmission. So, because they might have some plants that are infected but not symptomatic yet, and they're selling them and they're going who knows where. So that can be an area of concern. So if you choose to buy those plants, just know the risks, just be aware. So with the map though, you can zoom in and search by specific region or specific county, and it'll be up to date. So if you have any questions about that, I can send you Femi's email if you need it, and I'm sure he'd be more than happy to talk to you about it. I mean, I'm happy to talk to you about it, but he's my go-to person for this. All right, so Xylella is another insect-transmitted bacteria that also affects a lot of our high-value plants that require a lot of effort to manage and get established. But this group of bacteria causes Pierce's disease on grape. It causes pecan, olive, and shade tree scorch. I actually just had a sample of an oak tree that had bacterial leaf scorch. So it is out and about in our area of Texas. This particular pathogen lives in the plant vascular tissue and the insect, just like the last one we talked about. This does not survive very well out in the environment on its own, similar to citrus greening. So with management, we focus on vector control, clean graft stock, particularly with things that we get from cuttings like grapes and those woody specialty crops. And then we also focus on removing infected plants. And you can see one here in the photo. It's kind of in the middle. It's the one that's really small compared to the counterparts, and it's got that scorchy looking symptom. And if I were to just leave that grapevine there, it would just be a point of infection or source of inoculum or pathogen for all the neighbor plants. So we would have insect vectors, in this case, glassy winged sharpshooters, this little guy, which are also super tiny, go and feed on these leaves, get that bacteria in their mouth part, go to neighbor plants or even 10, 20, 30 feet down the row or across the vineyard, feed on a new plant and spread it. So it's really tricky to control the vector again because they're so small and they do travel on the wind. Currently, other treatments are being researched, and I think that the main times I've seen real effort put into vector control significantly in terms of like expense and labor would be for high value plants, like a heirloom plant or historically important plant, something like that. But it, again, is really challenging. All right, so this is the last group of bacteria. It's insect aggravated. Bacterial soft rots are very common in our vegetable production, unfortunately. These type of bacteria all make enzymes that degrade plant tissues, specifically cellulose, which is the material that gives plant cells their rigid structure. So if we lose that rigid structure, it's mush. And that's exactly what happens with soft rot. I see this on everything from peppers to cabbage to onions. It's very prolific. I most often see this late season and post-harvest. And the real struggle with this is that if you have one or two that are contaminated when it goes into storage or processing, you'll come out with a lot more than one or two that have been contaminated, particularly in the case of things that are stored for a long time, bunched together like carrots, onions, even cabbages. Those can have a really long shelf life in the refrigerator or in a dry, cool place. So it's a very long period of time for that bacteria to not only have its essentially epidemic, but it has more of a window to contaminate other items of produce in its storage location, if that makes sense. So with this, my standard questions that I always have at least one person in the class ask is, if you eat this, will it make you sick? I hate to tell you this, but you've probably already eaten it. So it's not uncommon. But I mean, my grandma, like, she used to just cut the funky part of the cabbage off or the carrot or whatever and use the rest, and we're all fine. That said, I'm not a medical doctor, but um, you know, it's very prolific in the environment, so you've probably already been exposed. So don't worry if you've unknowingly or knowingly consumed some of this. Now, in terms of prevention, we want to focus on preventing insect injury later in the growing season in particular, but throughout the whole growing season. And we really want to focus on strict culling or removal of symptomatic plants post-harvest before it's going into storage. Because if it goes into storage with others, it will contaminate them all, and then you'll have a whole batch that's spoiled. So, all right, on to viruses. These are submicroscopic. Many are spread by insect vectors. Some can be spread by mechanical means, like on your hands or on tools, just through physical contact. And some can be spread by seed. Oftentimes we'll see yellowing, loss of vigor, the death of the plant in some cases, and some classic virus symptoms include spots, like you can see here in the bottom here, mottling and malformed growth, like you can see here in the center. And then this last one is extra tricky, just kind of diffused yellowing. And sometimes it can look a little bit like a nutrient deficiency just in passing, but if you look more closely at the pattern, like here on this bramble, if you look at some of these leaves and kind of divide them down the mid vein and compare the two sides, it is not uniform on both sides. So it's asymmetrical. So looking at that, I would think, that looks a little virusy, but that's due to practice. So again, pattern can be telling, but not necessarily always 100%. That would just be enough of a red flag that I would want it to be tested with some test strips or with some DNA or RNA fingerprinting. All right, so we do have a little small topic to cover about virus naming. So it can be a little confusing when you're looking at virus names, and you might have come across this in like seed catalogs or on the little tags when you buy certain plants. Like on tomatoes, you'll see tobacco mosaic virus resistant, for example. Well, why would you care if it's tobacco mosaic virus resistant? Well, that's because a lot of viruses infect more than just the plant they're named after. So like on this peanut leaf on the bottom left, it has tomato spotted wilt virus, but the tomato above it also has tomato spotted wilt virus. So it's a classic example of a single virus can infect many different plants. In some cases, hundreds of plants, like tobacco mosaic virus can infect hundreds and hundreds of plants. So the way that the naming system works is the crop it was first found on, the symptom, and then virus. So it's just kind of a rough formula that was established a long time ago, and it's just stuck around. So it can be a little confusing. Prevention tips for viruses is prevent initial infections through vector control, using clean stock, tool sanitation, and remove affected plants. So a little case study. Tulip mania was a pretty substantial event in the 17th century in the Netherlands. And in this particular event, there were these really striking tulips that had this streaky pattern on their petals and on their leaves, and people just absolutely loved them. And they became very, very expensive, and people would trade futures on them and pay just huge sums of money for just a few bulbs, like enough to buy a car, maybe not now, but a car like 15 years ago, but still a huge sum of money for flower bulbs. And eventually, after a few generations, they would just kind of die out and have a lack of vigor, and they couldn't propagate them any longer. And no one could figure out why. Well, in the 1950s, it was actually recognized as a virus. And nowadays, our streaky tulips are due to breeding, not to a virus. Or at least commercial varieties that look like that are due to breeding. So with this particular virus, it is a good example of a plant disease affecting economics on a very large scale. I'm sure it had some social implications, and it impacted the lives of individuals in ways that were never really measured. But it's a good example of how impactful plant diseases can be. And it's less morbid than like the Irish potato famine and the wheat rust famines of ancient Rome and things like that. Because as far as I know, no one like died from this. So it's not as bad. But before I said that we can never really see a virus, and that is true without the use of highly specialized microscopy. So this picture on the right is actually a picture of some tulip breaking virus particles taken with an electron microscope. So an electron microscope is a very, very specialized microscope. It can do a magnification on an order of like 100 times what my best microscope in my lab can do. So we're talking like the highest mine in the lab could do, which is a basic like light microscope, could do like 1200 times magnification. For a really high end one, you could maybe do like 15. Well, not really high end, high-ish end one. So 1500 magnification, right? An electron microscope is more like at a couple zeros, like 150,000 times magnification. You can do anywhere from like 30,000 to I think like 180-ish. That number is pretty old in my memory, but that sounds about right. But basically, it's on a magnitude of several times over what you can achieve with electron microscopy. So you can actually see virus particles. They're very, very, very small, but only with very special equipment and special training to use that equipment. So these virus particles, though, are actually very cool. They are kind of shaped like pinwheels. When you look at them dead on, they're actually long tubes, kind of like this. And it's like a coil of proteins that are just wrapped around this tube. And then in the middle is the genetic material. So it's like a long coiled tube with proteins and genetic material, and that's it. They're not overly, they're complex, but they're not like complex like a multicellular organism, but they're pretty cool. So super neat stuff. So, oh, one other thing. So one thing to kind of like link together here is that in the 1950s was when electron microscopy really became more prevalent, and more universities and research institutions had electron microscopes. And that also went hand in hand with the time when a lot of plant virology and human virology really exploded with their knowledge of virus structure and also protein structures and things like that. So as we have these big leaps in technology, they often coincide with big leaps in our understanding and progress in a field of study like virology, for example. All right, so an example a little closer to home is rosette virus. This is characterized by discoloration of the leaves, also overproduction of malformed thorns. So those are really straight and they should be curved more like a cat claw. There's also way too many and they're too close. Node length can be too short, or in some cases too long, but in rosette it would be too short. You can also have overproliferation of buds. So instead of 1 or 2, you might have like 20 or 30, and they'll be really small and twisted, and they might not open, or if they do, they'll be just malformed little flowers that are have the wrong number of petals relative to the variety that it is. So a lot of growth malformation and discoloration. So this is spread very effectively by mites, like spider mites, feeding on an infested plant, going to a new one and feeding on that. Also not sanitizing your tools between plants when you're pruning. So if you're doing just regular prunes or cut flowers and you go from plant to plant to plant, you can spread it plant to plant to plant. Also physical disturbance of the plant, so like rubbing the leaves or picking flowers, and you get that sap on your hands and then you go handle other plants, you can spread it that way. Now, the order of those methods of spread is how effective it is in spreading. So mites spread it most effectively. Not sanitizing your tools, second most effectively. And then physical disturbance like handling is third most effectively. But by far, mites are the most effective means of spread, so much so that we actually use that in experiments. So we have little colonies of mites that we use to spread pathogens in experimental settings. So with rose rosette, though, it will always result in plant decline and death. So we really focus on prevention because we don't have any good treatments. We start with clean stock, space your plants so they don't touch to make it harder for those spider mites to go plant to plant to plant. If they're too close and the leaves overlap, it's like a little highway. You're just making it easy for them. So space them apart where the mites have to work for it. Sanitize your tools, remove any wild multiflora roses. You have less there. And some of the other areas that I give lectures for, like in North Texas, up by Dallas, they're prolific. And actually, where I grew up in Arkansas, the wild roses are bad enough you use a brush hog to whack them down for the year because people used to plant them as fences. So they're just this huge tangle of roses. So thankfully, you don't have it that bad there. Most of the issues I've seen around there are going to be more like your naturalized from old homesteads, but those are relatively few and far between. Also, cull your affected plants as soon as possible because you don't want it to just sit there as a little repository for the pathogen to spread to everything else around it. Here's another shot of a virus in ornamentals. This is tobacco mosaic virus on petunia. And this is a good visual representation of the infection timeline. So look at the leaves way down here at the bottom. They don't look particularly symptomatic versus the ones up here. The new growth is more symptomatic. So I would suspect that possibly this little plant was infected after it had grown those initial leaves. So you can sometimes use that as a visual gauge. It's more obvious, and I would be more confident in saying that about this rose. We're here on the lower area, you see all this normal growth, and then you have this little upshoot of malformed growth. And so people always wonder in my classes, oh, well, can I just prune that out and save the plant? No, unfortunately not. So the point of infection was earlier than this growth. So the point of infection is actually down here in the main bush. And by the time you're seeing this main growth, that virus is already proliferated throughout that tissue and put on new growth from infected tissue. So the most visually striking symptoms are after it's been infected. So we've had time progress. So it's already in the plant beyond that new growth, right? So it's kind of like an iceberg. What you're seeing is only a little bit. All right, so some common issues with viruses I see here in Texas are vegetable viruses, particularly in tomatoes and peppers. There's over 12 in tomatoes alone. Unfortunately, they're very prevalent. And I think that's due to we have a really long season for our insect vectors like whiteflies and thrips. And we just get really, really high populations of all of those. So they spread viruses very well. And a lot of these viruses also infect many different types of plants. So we can have very high concentrations of these virus populations in certain areas. There's unfortunately, again, no treatment or cure for viruses. Once it's in there, it's in there. We can't really fix it. So we focus on prevention of spread through managing our insect vectors, being mindful with your own sanitation, on your hands and your tools, and in some cases, soil. There are certain viruses that I would recommend, like pasteurizing your potting soil or something like that, but that's not the norm for viruses. Typically, we focus primarily on insect and human spread. All right, on to nematodes. So these are microscopic worms. Well, don't want to get ahead of myself. Okay, so they're microscopic worms. Only a small portion of them are plant pathogenic. So there are many, many nematode species out there in the world living their life, doing their thing, and are really important to soil microflora. There are many bacteria feeders and insect larva feeders in the soil that are not bothering our plants whatsoever. There's also a lot of parasites, like animal parasites, that are nematodes. So that's the less glamorous end of nematodes, I guess. But for plant pathogens, though, we focus on the ones that infect plants and feed on nutrients. Most of those that we encounter are going to be root feeders. And all plant pathogenic nematodes have a special mouth part called a stylet, which we can see in this photo right here on the bottom left. And here's an up close shot of that. So this little stylet is like a little straw that it can actually poke out to pierce the plant tissue, and it takes in nutrients that way. And it also secretes some saliva-like substances into the plant, kind of like a mosquito does when they bite you. It's not just slurping out your blood, but it's actually secreting some compounds back into you. It's kind of like that. So one of the most well-known nematodes is root knot nematode, which you can see here on this carrot and here on pecan. And there are several types of nematodes here in Texas that can cause problems on plants. Your prevalence depends on your soil type. Some nematodes really like it sandy, some really like clay-heavy soil. Some species have a really wide host range, some are very specific. So they're just highly varied. And many different types of nematodes have different feeding habits. For example, we have migratory nematodes and sedentary nematodes. So during their feeding stage, migratory nematodes will feed on a spot, move over, feed on a spot, move over, feed on a spot again. So they make a lot of little individual wounds, but they won't ever form a permanent site and form a gall like a root knot nematode. So a root knot nematode, in contrast, is a sedentary nematode. So its feeding stage is its feeding stage will form a sedentary site, basically, where it forms a little feeding site and stays there forever, or for its life. So that's the difference between migratory and sedentary. So let's think about that, how their life cycle is going to have a very different impact on the type of damage that's done to the host. So in migratory, we have a lot of little different wounds, versus sedentary, we have one original wound, and then we have this malformed growth. And that malformed growth is actually going to be made of a few different components, which we can see here in the early stages of development on this bottom photo. So G right there is a giant cell, which is the initial kind of feeding site. So a root knot nematode, when it picks its little feeding site and it pierces the plant, it secretes these compounds that basically tell these cells, it kind of hijacks their metabolism, and it tells them to crank out a ton of sugars and other food products for the nematode. And so that cell starts growing larger, and just it kind of makes its own little food supply right there. So that nematode has its feeding site with a giant cell. So that gall is going to be part giant cell. And then the mature female, which females are the only ones that are actually going to feed on the plants, the females will form an egg mass. So part of that gall is going to be egg mass, which is EM in that photo. And you can actually see the little white kind of like cylinder-shaped eggs within the brown part, and the brown part is going to be the female nematode's body. So that egg mass is going to be in her body. So that female nematode is going to complete the life cycle there, have some egg mass and a giant feeding cell, and that's going to be what makes up your galls. So very different type of damage to the roots than repeated feeding sites, right? So our life cycle has a big impact in how the damage is manifested. So that's the main thing to take away from that. So nematodes are really difficult to manage because there's just a lack of labeled products for homeowners, and honestly for a lot of commercial producers, particularly with vegetables. So we really focus on prevention, use resistant varieties, prevent infestation in the first place, so inspect all your transplants. Use crop rotation, being careful of not using alternative hosts. So let's say we have Meloidegon incognita, which is a type of root knot nematode. Well, if I plant tomatoes and then cucumbers and then like eggplant for my 3-year rotation, well, that didn't do any good because it can infect all of the host crops. So I've just continued their life cycle. So the point of crop rotation and using different types of crops in that particular site where we have a known pathogen is to essentially starve out, if you will, the pathogen. And if I continue using hosts that serve as a food source for it, then I'm not doing any good. So now we're going to dive into fungi. This is, again, the most common type that I see. The good news is most fungi out in the world are not pathogenic to plants. Most are just decomposers contributing to nutrient cycling. They're not like harmful to anything. They're just breaking down old dead materials. And there are some that are symbiotic in nature, like mycorrhizal fungi, which increase the root absorption zone area for nutrients and water. They also assist with breaking down compounds into more available formulations for plants. And they also the plants provide carbon to the fungi, which can then break it down and help store it in the soil as part of its biomass, which greatly assists with sequestering carbon from the atmosphere, right? So when we talk about like, oh, well, the rainforests are great for sequestering carbon and grasslands are great for sequestering carbon from our atmosphere, they are great, but they are only a small portion of the picture, because if they did not have mycorrhizal associations, they would only be able to store a fraction of what they actually do. So the mycorrhizal association is dramatically improving the effectiveness they're able to sequester carbon from the atmosphere. So it's super cool. They also facilitate signals to and from plants, like between plants, using chemical talk. So it's kind of like using the 2 cups in a string, but with chemicals, going through the mycelium or hyphae, which we'll look at some pictures of that in a minute. The hyphae, not the chemicals. So in a nutshell, fungi are super cool, complex, and most of them are not actually hurting anything, but some of them do, and that's what keeps me employed. So now, again, this is the group we usually see literally. The appearance of structures we see is going to be highly diverse and varied in size. Some are, the size of like a golf ball, like these jelly ears. Some are going to be microscopic, like bread mold. We can see bread mold, but their reproductive structures are actually microscopic. You know how when you have some really funky bread and you open the bread bag and like dust comes out? That's not really dust. Those are spores coming out. Just the structures that make them are so small, you need a microscope to see them, which you can see in the purple, or in the top photo, they've been stained purple to make them more visible. And that's around, I'd say, 550 times magnification for reference. Now, on the other end of the spectrum, you can have large structures like these bracket fungi, also called conchs. Those can be like 2-3 feet in diameter in some cases, and just monsters. So very, very variable. All right, so fungal structures broken down a little more. We have the mycelium or hyphae. This is the vegetative growth or the somatic tissue, which is the body of the fungus. Think of it kind of like roots. It colonizes the substrate, which may be soil, it may be plant material, it may be a dead critter on the road. Just whatever it's colonizing, that's its substrate. This is a sign. It's not always visible because sometimes it's down in the substrate. But like on this log where someone's peeled the bark back, you can actually see that top layer. Now, singularly, they're hyphae. Collectively, they're mycelium. And you can also see that in I think that's a little more familiar, a mushroom. So the mycelium, made up of hyphae, is below ground. The reproductive structure is also made of hyphae, but it has some specialized spore-making structures underneath the top side of it, and that's the reproductive part. So you only see the reproductive part sometimes, but the mycelium is there all the time. All right, so reproductive structures. Spores are the main one that you'll see out and about. And these are kind of like seeds in that their whole purpose is to go off from the parent, find a new place, land, and grow. Now, spores are very, very abundant. They're made by the millions, but only a small fraction of them actually land where they're supposed to and actually grow. They do vary greatly in their size and color, and we'll talk about that shortly. But once they do land somewhere, and if they have favorable conditions, they'll grow and branch off the hyphae that make up the mycelium and colonize their substrate. And in this case, on the bottom left, the green is the leaf or a leaf surface. That little opening is a stomata, and this little spore has germinated a little hyphae. Looks kind of like a little root, right? This little hyphae is going to try to sneak right in that stomata and gain access to the plant, and then try to overcome those chemical defenses and colonize that leaf and start its process of colonization and reproduction. Now, these center photos, you're probably like, what is that weird stuff? Well, that's actually photos of hyphae inside a stem on the top, and then the bottom photo is inside a leaf. So someone has cut them open and made a thin little layer, and that the string looking stuff, that's the hyphae that's embedded inside the plant. And on the bottom one, it's been dyed blue just to make it more like visible. So it's really down in there, kind of like roots. And its whole job is to absorb nutrients from its host, or in the case of non-pathogenic fungi, whatever material it's breaking down. All right, so spores, I touched on this before, but I got ahead of myself. Spores do vary greatly in their shape and color. Some are darkly colored, some are more clear. They can be teardrop shaped or canoe shaped, or just single, like a single little oval. So the characteristics of the spores are actually a very helpful feature in making a diagnosis in the lab. So if you send in a sample, a lot of times we'll take our little tweezers, pluck some tissue off, or culture it and then pluck some of that tissue and then put it under the microscope and look at these features, and that can tell us what exactly we're dealing with, and then we can give you a diagnosis. So how do these spores get from A to B? Well, there are a lot of different ways, but we'll start with something a little more familiar, like a mushroom. So on the underside of a mushroom cap, we have these little flappy things called gills. Flappy things is not the technical term, but I still like it. So these little gills are actually covered in these little structures that make spores, and there are millions and millions of spores made. And when they're mature, they drop off. And I actually have a video for you guys. Here we go. Okay, so this is the mushroom stalk, and then the cap is at the top, so we're kind of like a little fairy sitting underneath the mushroom. And this has been grown in straw, so that's the pokey stuff you're seeing. And all that dust-looking stuff blowing around, that's actually spores being released from the mature mushroom. So this doesn't have like a fan or anything. It's just normal room air currents, and you can see how effectively these spores are spread and just drift along on the air and can go off to find new locations. So this is a passive dispersal. So they're just kind of floating along. And this is, again, it's from YouTube. It's just spore rain. And I can send you the links. It'll be in your handouts. Alrighty. Okay, here we go. So keeping that in mind, let's look at something a little different. So on this bottom right, we have this structure that looks kind of like these horns poking out from a little ball of brown clay. Well, that little clay-looking ball is actually a reproductive structure along with the horn-looking structure. And those little appendages are actually covered in spores, and they emerge when that structure is mature. So this is a part of the life cycle of cedar apple rust. And thinking about where that's located and how those little horn structures have emerged, do we think that'll be water splash or wind spread? Given that it's in the middle of a tree, I'd say wind spread, right? Because it's kind of at a prime location to be caught up in the wind and then taken to an apple several miles away and then infect that apple, right? So the location and structure of reproductive physiology of fungi have a direct role in how their spores can be spread. Another very different one is anthracnose, and this is on the bottom left, a microscopic shot of the little reproductive structures. You can see they look like a little sea urchin kind of. I grew it in culture, and you can see that on the top left photo, and those little, it looks like black pebbles, those are the reproductive structures, and that goop on the top is actually spore ooze. So it's droplets of thousands and thousands of spores. And let's say we have water splash. And here's a little cross section of what that looks like because it's kind of hard to see. So it's kind of a cup shape. So if we have a little droplet of rain hit that and splash out, that's going to be very effective at spreading spores through water splash, right? But given that we have little droplets of moist spores, they're not going to be picked up by the wind very well, right? So understanding the physiology of the pathogen in question really helps us understand how we can take steps to prevent the spread. All right, so other survival structures. I know you've already heard about a lot of fungus stuff, but there's one last thing. Some fungi, particularly a lot of soil-borne ones, actually make these tough little structures called sclerotia, and they're like little pebble tough things. They're actually little bundles of modified hyphae, and they can last in the soil for decades and through really unfavorable environments. And we can see that here on this potato. So we see hyphae on it, those little webby things, and then these little chunks. Those are sclerotia. They vary in size from like smaller than a pinhead to, you know, maybe a 16th of an inch, maybe 18th of an inch. And here you can see it in culture, those little tiny white beige colored flecks. Those are baby sclerotia starting out. And if, say, this stem had been left in the field or that potato had been left in the ground and they rotted away, those little sclerotia would still be there years from now, if not decades from now, and able to cause disease on a new plant that happens to grow in that location. So several fungal diseases make these structures. Some of them can infect a really wide variety of hosts, and a lot of fungi that make these are soil-borne. So that's highly advantageous if you're bound to being in the soil. If you can make a structure that lets you live for decades just waiting for a plant to happen to grow there, that'd be really advantageous, right? All right, so cotton root rot is a very prolific issue in our area of Texas and throughout the Southwest US. I actually just found a new spot of this, well, new to me, last week in a cotton field. It also goes by the name Texas root rot or Phaumatotrichopsis root rot, but it is going to thrive in relatively high pH soil. It really likes like 7.5 to 8. It really likes hot soil temperatures over like 85 or so, and it can live for over 100 years in the soil, possibly longer. That's just as far back as we've recorded. And when I say it, I mean its sclerotia can live that long in the soil. It has a wide host range, ranging from cotton to grapefruit trees, a lot of our ornamentals and vegetables, over or upward of about 1,000 crops, I think. And we typically will see wilt and a scorched appearance followed by rapid death on infected plants. And the leaves will then stay attached. And the epidemic, if you will progress from a focal point. So it'll start in one spot in the field or the orchard and then just progress outward. It does spread primarily by mycelial growth through the soil, people moving soil, and well, those are the main two ways. There are no viable spores. So it does make these little reproductive structures. It's a little crusty mat on the ground that's around 4 to 6 inches wide. And it does make spores, but they're sterile. So that's a little quirk of this pathogen. So here's the up-close shot of what that looks like in peach. Notice the leaves have died. They're hanging on. And this tree completely died within, if I recall correctly, about 2 weeks, 3 weeks maybe. Here's a shot of cotton root rot in a field. This is actually from Nueces County, so pretty close to you guys. And this is why it takes me forever to go anywhere. I have to stop and take pictures. But it is a classic cotton root rot hotspot. I like to call them UFO spots because it looks like a UFO just came in and like scorched a whole spot. But that is just from cotton root rot. And you can really see how it's progressed from a focal point in the center. It is really difficult to manage because it can live in the soil for a really long time. So we focus on replanting with immune plants. Like a lot of our natives are tolerant to it. Monocots are tolerant. For commercial producers, we do have a fungicide labeled, but it's only approved for use on cotton, grapes, and hopefully soon to be olives. Currently, we don't have any soil tests we have examination of roots is the only means of confident diagnosis, like for sure. So you have to dig up the roots promptly before they start to decay, send in a sample, and we look for these little yarn-like structures, and we pluck them off and look at them under the microscope and look for these special little structures called cruciform hyphae. So they make a little plus sign. All right, so on to fungi-like. Oomycetes are water molds, and back in the day they used to be grouped with fungi, but in the past 20 years they've been broken out more or further away from the taxonomic or relatedness of true fungi. This includes downy mildew, and they have a very unique means of spore dispersal. We see a lot of damping off with oomycetes and root rots as well. And there's the classic downy mildew on spinach, which is a really big problem in the spinach production, both here and across the US and internationally. An oomycete I see a lot here in Texas is Phytophthora blight of pepper. It survives in the soil and in crop debris and maybe seedborne in some cases. And it's characterized by chocolatey colored to blackish rot on the stems, and it will progress from the bottom up usually. And this mummified looking fruit with like a water soaked lesion on the or margin on the edge. So we really focus on future replanting with resistant varieties, vigilant use of fungicides, and sanitation of infested material, because you don't want to leave stuff around to be a source of pathogen for next year, and clean seed, which is especially important if you're like saving seed or trading seed with a friend. Just be really vigilant about what type of fruits you save seed from. So a little closer look on oomycetes. So one of my first samples was hydroponically grown wheatgrass, and it was a pretty big operation. And they just had this problem where they had 90+% losses every week, and they brought me some. And I flipped it over, and the roots were all kind of brownish colored, and there was a ton of sign. So we had a lot of hyphae. Notice a lot of the terminology is going to be the same as we use for fungi, and it looks very similar until we look a little closer. But we have sign and symptom here. So I put some under the microscope, and I noticed these little things that look like little spheres embedded in the roots. So this is a closer shot of that root, and these parallelogram shapes are the cells of the root, and then these little round spheres filled with this gunk, for lack of a better term. Those are actually zoospores in there, but those are reproductive structures of the pathogen. So I have a quick little video. Okay, so these little zoospores start out in these little structures right here called sporangia. And when they're mature, they emerge. It's kind of like popping a cork on a champagne bottle. And so this is in a thin film of water, and these little zoospores are able to swim around like little tadpoles. They do have 2 flagella. They're just clear and we can't really see them. So in a hydroponic setup where we have water circulating every 4 hours, I'm sure you can imagine how this became a really big problem very fast, because zoospores love water. So when we have like waterlogged conditions, I'm sure you can think of how severe this can get pretty quickly, like if we have some soggy peppers in our garden. Okay, so once they emerge, these zoospores don't just like randomly hang out. They actually have a process that helps them hone in on their basically food source or their host. So that is called chemotaxis, and basically these little zoospores can sense the compounds that the roots release. So sugars, growth hormones, and it's strongest at the root tip, like the actively growing region at the tip. So they're swimming around in water. We're going to skip along. So here's our little root tip, and these zoospores are able to hone in on that because it's a really strong chemical concentration that they can sense. And once they get there, they actually will shift gears dramatically, and they will insist, not like put your foot down, but like ENCYST, insist, like encapsulate. And then they have some pretty big metabolic changes, and they they will actually put out a little germination tube or hyphae, and that will penetrate the plant and then start the infection process within the plant. So we go from a free-swimming, mobile zoospore to acting a little more like a traditional spore or a conventional spore, right? Pretty wild stuff. So oomycetes are very different from true fungi on a, on a physiological and metabolic level. And what does that mean for us? Why do we care? Because some products, like fungicides, are only labeled for oomycetes. Some are only labeled for true fungi. Some other products are labeled for both. You just need to be aware they're different so that if you need to apply fungicide, you understand why the labels are written as they are. Also, be aware of the life cycle differences between true fungi and oomycetes with regard to management efforts. All right, so this leads into why do we care about getting accurate diagnosis? Well, incorrect management attempts make your problems worse and waste your time and money. And again, most fungicides only work on their specified labeled pathogens. Also, you need to know what you're dealing with. Is there an insect vector in play? Not all the pathogens we talked about today have insect vectors that we need to worry about. Some do. Also, do we need to worry about water splash? Do we not? And also, it's good to know, do you need to practice informal quarantine and sanitation of debris, and how vigilant you need to be on your own garden site. So steps to diagnosis include identification of the host plant, assessing your problem, doing your research and developing a suspect list, and narrowing the list of suspects and get help as needed. And again, this is just to give you guys an idea of the process so you better understand what information to bring to specialists and your county agents. You're not going to be expected to actually diagnose. So ID that host plant. It's important to understand this so you can automatically eliminate a lot of potential possibilities. And there's some really great resources, but beware of common names because they're not regionally universal. Assessing the problem is examining the whole plant, top to bottom, and the surrounding area. Ask questions about the site history, like weather events, any sprays or drift potential, and how long has this happened? Generally, if it was seemingly overnight, that's going to be an abiotic problem. If it took time to progress, then it's going to be a pathogen. And how many plants are affected? Is it a single species? Is it a lot? How's the area distributed? Is it a geometric pattern? Are there sharp angles? Those are kind of good clues about if we're dealing with a man-made issue or a pathogen issue. Now, this is the tricky part where we have to evaluate those symptoms and sift through a lot of information and develop kind of a suspect list. Signs are a really good tool at this stage, and utilizing really reputable resources, unregulated blogs and websites are iffy at best. I also like to consider what's common on this plant host. More often than not, that's going to be what you're dealing with. Part of my job is to watch for things that are not common and be vigilant for that. But most of the time, it's business as usual, which is not a bad thing. Lastly, narrowing that list of suspects and coming up with a specific diagnosis. It can be tedious and requires a lot more expertise of techniques and just general practical knowledge. But a really good primer set to start with that I cut my teeth on are the Compendium series. There's a whole variety of them on different crops, and there's tons of color photos in them and individual write-ups for specific diseases and problems, not just pathogens, but insect issues and abiotic problems. They're wonderful, but they're not cheap. They range from $50 to $150 on average. Digital copies are a little bit cheaper. And full disclosure, I do not get any benefit from telling you guys about these. I don't get any sort of money from them. I just really like them, and they were instrumental in me getting my feet under me when I was a grad student. So if there's a particular crop you like, just maybe buy that one book as a class book for everyone. So, you don't have to suffer from sticker shock for the whole collection. All right, so when y'all are master gardeners, people are going to bring you stuff and they're going to ask you things like, let's say someone brings you leaves, a lot of different species. They all have a rough leathery texture and it's on a lot of different plants and there's general yellowing. The first thing that you would do would be ask about the host ID. So in this case, the lady brought me a bunch of citrus branches. The second was assess the problem. I took a site history, asked her what had been going on, had there been any sprays, how extensive was this issue, and come to find out, this was actually a severe mite infestation. So it's a little bit of a trick question. It wasn't actually something that was truly pathogenic in nature, but it's still stuff that you will be asked about and that I'm asked about a lot. All right, so case study 2. Let's say a dying plant's brought to you, like the whole thing. It has defoliation and severe wilting. What's the first thing that I ask the person and what would I first do to the plant? And this is what I was brought. So some poorly looking plants, lack of vigor compared to a normal plant on the right. So first question was, what is this? I had never seen these before, and this is actually sesame. We grow a lot of sesame in Texas. And so I inspected these plants, took note of the lack of vigor, but then I noticed down toward the soil line, we had these deep sunken lesions. So I wanted to investigate a little more and I cut them open and saw a lot of discoloration inside. And I was a little suspicious that this was Rhizoctonia and maybe some other issues that I dealt with on cotton in Arkansas during grad school. And I cultured it and found out it was Rhizoctonia and charcoal root rot. So this is a really good example that plant pathology is not always just a one and done deal. Sometimes we have multiple things to play and they're extra tricky to diagnose without proper lab techniques. All right, so we're going to go through disease management. And again, just a reminder of these two topics, the disease triangle and disease cycle. There are points on each of these where you can try to basically break the cycle and disrupt the disease epidemic or prevent it from happening. So the best one is resistance in immune plants, avoidance through cultivation techniques, excluding the pathogen, protecting the plants, eradication of the pathogen, and therapy. These first ones focus on prevention and the last two on treatment. Resistance and immunity is exactly what it sounds like. We're using genetics and traits of the plant to naturally resist pathogens. It's super effective, as you can see in these photos. These were both, or in this photo, these were both exposed to the same amount of pathogen, the same conditions. One's looking great, the other one not so much. Now with resistance, we have direct resistance, which is when we have the genetics of the plant allow it to have a better chemical defense system, basically, and it can recognize that pathogen and fight it off better. Indirect is when traits that just happen to be there, like a thick waxy cuticle, can help prevent pathogens from gaining entry, for example, but it also helps prevent excessive water loss, sun scald, sandblasting. So it's not just there for disease resistance. Avoidance is manipulating your environment to be unfavorable for the pathogen and favorable for your plant. So adjusting your irrigation, avoid sprinkler use, using mulch to reduce the frequency of water, tillage to help turn under any crop debris that's left on the surface to help that decompose faster. Change up your planting site to make it less favorable. We don't want things like that middle photo where we have soggy plants. And changing up your planting time. For example, if like with cotton root rot, it really, really likes it hot. And cotton farmers used to plant extra early to try to harvest their cotton before cotton root rot really got going in late summer. So there are some tools like that that are really great. Also, a big one is use a variety adapted to your area to avoid plant stress. If you try to grow something that's really not meant for your environment, that poor little thing is going to be doing all it can just to live. It is not set up for success. So try to pick something that is adapted for your area. The Texas Superstars are great. There are a lot of varieties the Hort department have spent a lot of time developing and working on. So there's a lot of options. Exclusion is keeping the pathogen out, using only healthy plants. This is really important if you're going to be investing a lot of money on setting something up like a citrus grove or a vineyard. Maintain clean equipment, and that includes infrastructure to maintain a clean growing area. For me, that means have all my stuff ready to go to disinfect my tools, because if I don't have it right there, I'm not going to do it. The limitation is you can't control everything. You can't stop the wind. It happens, but it's all about stacking the deck in your favor for success. Protection is using a barrier to protect your plants from the pathogen. This can be a chemical barrier, like a fungicide, a biological barrier, or a physical barrier. So it's kind of like a little invisible raincoat when you apply a chemical fungicide or biological barrier. The mechanism is just different for each one. So biologics can be a living microbe that you apply that then colonizes your plant or your plant roots and the soil, or it can be a microbial metabolite or extract, basically, that you apply that is harmful or a deterrent to the growth of pathogenic microbes. A fungicide is going to be a deterrent to like fungal pathogens, for example. So it's all about just having like a little invisible cloak on your plants. Now, with time out in the weather and everything, that does wear off, so you have to reapply. Now, a physical barrier is just physically putting your plants in a different spot. Let's say you have an heirloom tomato that's super susceptible to root knot nematode, and you really want to plant it, but you have root knot in your garden. So I would just recommend plant it in a raised bed or a container. Problem solved. So it can be a really simple solution for what seems like a really big problem. So those are all great options. Eradication is getting rid of the pathogen or killing it. You can do this by physically removing diseased plant debris. Used to, people would fumigate the area and pair that with solarization, which is putting plastic down to heat up your soil. Nowadays, people try biofumigating with brassicas and then pair that with solarization. And if you're interested in that, shoot me an email. I actually did my master's on that, so I'd love to talk to you about it. I also have some fact sheets about solarization if you're interested in that as well. But it's a great way to kill out some soil-borne pathogens and get rid of a lot of your weed seed bed. Therapy is helping the plant to heal, and you're going to be utilizing multiple techniques like systemic chemicals paired with pruning. You don't ever want to rely on one technique alone, because that's not going to be very likely to yield success, like if you use multiple methods. So I'm a big proponent of use more than one method for control, and you'll have a much better outcome. IPM, in a nutshell, is just using multiple disease control approaches to prevent and manage disease, and it needs to be multifaceted, ecologically responsible. I like to throw in fiscally responsible, and evidence-based, and have a heavy focus on prevention. So here's some home reading for you guys. It's just kind of a little walkthrough on an IPM situation. Other problems include herbicide damage and weather issues. We've all seen that the past couple of years, like freeze damage and drought stress. Soil salinity, especially in repeat irrigated situations, especially potted plants. We get that a lot. If you have well water, I see a lot of that problem in potted plants. And fertilizer burns are really, really common, especially with like foliar application fertilizers that are incorrectly mixed. And there's a close-up of an herbicide burn on a citrus leaf. And note how that looks different than the leaf spots we looked at before. We have a very clean edge. The middle is not colonized with fungus. And if we were to check that again in like a week, it would be the same size. It would not be progressing. So that's a key difference. Fungal issues and other pathogenic issues progress. Arthropod pests, again, are super common in our area. They cause so many problems. Everything from malformed growth to just general decline. They also spread a lot of pathogens. Whatever you choose to do for management, stay on schedule, because if you skip applications, you're encouraging pesticide-resistant populations of insects to breed and populate. Molly Keck is wonderful. She is an IPM entomologist in San Antonio. She is fantastic. And if you have any questions, I highly encourage you to get in touch with her. You can Google her, or I can send you her email. Sooty mold is not really a plant parasitic fungus, but something I get a call about a lot. It's actually a fungus that grows just on the surface, eating insect secretion, so insect poo, basically, also known as honeydew. This will resolve and completely go away once you get your insect problem under control. It looks gross, though. Parasitic plants are really problematic. I didn't see dodder, which is this kind of yellowy orangey vine, for years living down here. And then the past couple of years, we've actually had it pop up in the springtime, and then it dies off once it gets really hot. So be on the lookout for dodder. It penetrates the host, so it's a true parasite, and the only good way to remove it is just physical removal. There are some herbicides labeled, I believe, but you're going to be sacrificing the host plant. So, proceed as appropriate for your situation. Mistletoe is another true parasite. This is a major problem in our area throughout pretty much all of Texas, especially when we have stressed trees. It gets like a little foothold, and then it's just like wildfire and takes the tree out even faster. And again, it's a true parasite that actually penetrates into the plant. So even if you remove the green part of the mistletoe, it's going to pop back out because you didn't get all of it. Mossballs are thrown in here because they're often confused for parasitic plants, but they're not. They're actually epiphytes. They're in the same genus as those fancy little air plants that we all buy because they're cute. But some people don't like the way they look, so you can treat them with copper or potassium bicarb. Baking soda is sodium bicarb. You can use that, but it's not going to be as effective as potassium bicarb. All right, so resources. Kevin Ong is in College Station. He's great. He's the director of the plant clinic. I'm in Uvalde. Femi Lobby is in Westlaco, so you guys are right smack between myself and Femi. And there are a ton of instructional videos on the Texas Plant Disease Diagnostic Lab website. And they also have a Facebook page that has a lot of really cool posts and What Wednesday programs. And then I have a Facebook and Instagram under Plant Dr. Kim, and it has everything from plant pathogens to just weird looking plants that I come across to the random stray emu that lives here in Uvalde. So you never know what you're going to get on my page, but I guarantee it will be entertaining. Here's some sample shipping tips. The main thing is don't add water because it does not travel well. And here's some resource websites. The Plant Disease Handbook is a continually ongoing effort of overhauling. APSnet.org has some material that you have to pay for, but some of the learning content is free, and it has some really good, well-reviewed, thoroughly vetted stuff. This is the main organization for my professional group, so plant pathologists. And trust me, if it is wrong, someone will tell you. Now, for extension, there's the soil testing lab. I don't know if you guys have heard of it, but it is great. They have really good prices. I use them for my experiments. The Uvalde Center here and the Plant Clinic website is here. If you're interested in hearing about oak wilt, there's a great website for that. And the Forestry Service has some really great tools on tree selection for your specific area. You can put in your zip code and the characteristics you want about a certain tree, and it'll spit out a bunch of options. So it's really cool. The Texas IPM program has a variety of content ranging from mouse management to insect management to some plant disease stuff. UC Davis has a really great IPM program as well with a lot of really good pictures. And I mentioned that there were a lot of cotton root rot susceptible crops and then some tolerant native crops. And here's two lists that have that information on there. And then if the microbiology aspect of this course kind of piqued your interest, Cliff Notes actually has a really interesting little free tidbit on microbiology in general. And there's a little home activity for fun. Mushroom spore prints are a thing. We actually do them to characterize mushrooms for herbarium samples, but we also, nerds like myself, we all like to do them as little art prints and things like that, so they can be fun. And there's my contact info, and that is it. So you guys, do you have any questions? I know we're right at time. We're like 2 minutes over. 6 minutes over. But I have time for questions if anyone has any.
Thank you, Dr. Cochran. We appreciate it. You always do a very thorough job, and we appreciate you taking your time on a Monday night to teach us.
Oh, I do not mind at all. Well, thank you guys for having me. You're always a good group. I know it's hard online because it's not as engaging as in person, but it does offer a lot more flexibility for everybody.
Yes, ma'am. Are there any questions for Dr. Cochran before we let her go and sign off?
If you have a question, don't forget to unmute because you're muted. Remember, guys? I guess everybody's so fascinated they can't think of any questions.
I'm going to assume I did such a good job. No one has any questions.
They're saying thank you, though, in the chat. Thank you, Dr. Cochran. I enjoyed it because I like diseases and plant problems.
Oh, yeah. The thing is, like, every single plant pathologist is a massive nerd. So if you get us talking about the things that we like to nerd out about, we'll be here all night. So if y'all have any questions that you think of later, feel free to email me. Don't be shy. I love to talk shop.
Make sure to show somebody that picture of that plumeria I sent you.
I will. And I actually might bug you for some like high quality photos if you have them.
Okay. Yeah,
like if
I
that
have.
plant is still in place, maybe some more up close photos.
Yeah, I think I'm going to take a sample off of mine because I have it here. Heck, I'll just mail you a whole branch over there. But no, I want to cut mine open like one of those little pieces and look at it.
Oh, yeah. Yeah, for
just
sure.
to see what it has in there because I just discovered today that I have a cutting of that plant. So
nice.
So all the interns are telling you thank you. They said great presentation.
Awesome.
Thank you so much.